This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 214, recorded on March 26th, 2020. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, how do you do? How are you in these strange times?
Aren't they strange, though? It's amazing.
Boy.
A virus can change the world so
rapidly.
And also from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
I take it you both are healthy, right?
Yeah, we're doing fine.
I'm taking my temperature 3 times a day as prescribed by my employer. So,
But you're not going out, Michael, are you?
No. I had to nip into work a couple 4 times last week to do some press interviews for the local media on behalf of my infectious disease colleagues who have been doing yeoman's work getting the hospital ready for the surge that you are seeing in New York City right now. We're anticipating it's going to come to Charleston soon.
Yeah. Our hospital yesterday had over 800 patients, COVID patients.
800 COVID patients? Wow.
And they said half of all admissions are COVID positive.
Wow.
Good grief.
And they're using 50,000 face masks a day.
That's up 10,000 from last week. Your chief of surgery published that he was using 40,000 last week. So that's a net increase over what you had said last week. So that's pretty scary.
Yeah, they're going to have a problem if they don't get some. So, Elio, are you staying home also?
Yes, sir. I live in a senior residence. They take very good care of us. They're bringing us the food, and we're not supposed to go out, and I do go out a little bit to get a breath of air, but I don't touch anything. I don't see anybody. I stay away more than 6 feet.
Okay.
So far, so good.
Yeah, I went to work on Tuesday because I had to change the liquid nitrogen and the carbon dioxide tanks.
Oh, the jealous mistress.
So I went in for the day, and it's pretty deserted. The city is very empty, but the streets are not completely empty. There are people walking around. There are kids on the playgrounds,
Mm.
But I've been in my basement for the duration. So I want to ask both of you, how long do you think we're going to be in this situation where we can't go anywhere?
Well, that's a broad question, isn't it?
Yeah.
The models that our chief hospitalist has posted today is they're anticipating 8 more weeks of this here in Charleston.
In Charleston. Have you gotten started yet or not yet?
We are seeing a significant increase. The first cases that we had in South Carolina were in Kershaw County, but our hospital was only stood up its own testing program on Monday.
Mm-hmm.
So we have had a rough time getting set up to do testing, and I think that's what really why New York City is so high. It's because you guys were so ahead of the rest of the nation in being able to test folks. We're just all behind, yeah. I think.
What do you think, Elio?
Well, I am wondering, in general, not just locally, it seems to me the first thing to come online will be drugs, and vaccine much, much later.
Mm-hmm.
So being that there's some 60 different drugs are being tested, it would seem incredible that some won't work. And so would they be prophylactically useful or just therapeutically? Who knows? Yeah. But it seems to me that before long, we're going to have effective drugs. And when we do, it's going to change the equation. What do you think?
I agree with you on that. And the number of drugs being investigated is now up over 100 as of today. So they added 40 new compounds since you last updated. And I think the number are just increasing with each passing day.
Where do you get that number from, Michael? Is there a place I can look?
It was a web posting that I grabbed. I'll send you the reference.
Okay.
Because when I was writing up the results that we're going to talk about in the snippet, I stumbled across it.
So that's the optimistic view.
I don't know. I think 8 weeks is a long time. I think the people can't stay home for 8 more weeks, essentially. I think that would be a real problem beyond the virus. Now, I saw some numbers yesterday that the outbreak in New Rochelle here in New York is going down, which is an interesting observation. And so I think in some places when we see that going down, I think we have to bite the bullet and get people back, take a risk that we may have reinfections, because I think the damage can be worse from everyone sitting at home for 2 months.
Yeah. The only people who will benefit are divorce attorneys.
The thing is, one more thing I wanted to just run by you both. I mean, have we ever stayed home for any infectious disease outbreak?
Well, Spanish flu.
Did people stay home for that? Yeah.
That was not in my lifetime. So I was going to say not in my lifetime. Even the pandemics of 1967, the last big pandemic flu, we didn't stay home.
No. The
pandemic of 2009, the scare in 1976 over swine flu. I mean, this is precedent setting. We have never seen anything like this in the modern times.
Elio, were you around for the 1918 flu?
No, sir.
It was a glimmer in his father's eye.
I thought you were.
It was not even that. No. I was born 10 years later.
Okay. I'm sorry. I didn't mean to say you were old, but.
Well, it turns out I'm quite old. Old I am.
Yeah, but you do very well on TWiM, so good for you.
Thank you very much. Well, trying to stay up on things. Anyhow, that's actually a pleasure. I'm glad I get to do them.
Well, thank you. We like it very much. All right, so we do have a paper for you, but let's hear. Michael has an interesting snippet which has to do with the virus. So tell us what's up, Michael.
So some of you may know this year I've had the privilege of serving as the chair of the Council of Microbial Sciences, which is an advisory body to the American Society for Microbiology's board of directors. We, along with the American Academy for Microbiology, are the board's, if you will, think tank for all things microbial. It's a big group with lots of expertise and insight, where we're comprised of 35 branch counselors, 27 divisional counselors representing all the scientific disciplines of the field of microbiology, immunology, virology. We also have the 8 program chairs that effectively manage the ASM, as well as 11 at-large counselors, and then 7 ex officio members that are from the board, bringing the grand total of this think tank, if you will, to 88 really bright individuals. In late February, I was talking with our chair-elect of comms, Dr. Suzanne Fleischig, a former TWiM guest, episode 56, if you're trying to remember when you heard Susie last, where the topic of SARS-CoV-2 or COVID-19 came up, and this virus will likely touch all of us, every human on our globe this year in some way or the other. And during our conversation where we were discussing issues affecting comms, we thought if there was ever a time for comms to mobilize, now was it. So collectively, we wrote to the leadership of the society, Dr. Robin Patel as president and Stefano Bertuzzi as the executive director, and we offered that comms was ready to convene a global summit in a short period of time. So this was actually February 27th. And so we suggested to convene a global summit on this COVID-19, specifically where comms would marshal its collective expertise and that of the broader ASM membership and its staff to help to contribute to the scientific efforts aimed at addressing this global threat. And rather than reviewing results that are being deposited daily within the peer-reviewed literature and the preprint servers like bioarchives, medRxives, and chemarchives, we would assemble a group of experts from the diagnostics and epidemiology realm, the therapeutics, / fundamental virologists who could actually contribute to where the science needed to focus on from the development of therapeutics. And finally, the area of prevention, which not only covers vaccine development, but as well as this whole notion of social distancing. Now, Dr. Fleisig and I had no illusions we could do this by ourselves, but rather we needed to tap on experts from comms. So we asked Dr. John Parker from Cornell, who is the counselor from Division T, aptly named RNA viruses, and Dr. Esther Babity, who is the branch counselor from the New York City branch. And Esther happens to be the chief microbiologist running the diagnostic laboratory for Memorial Sloan Kettering. So she has tremendous expertise in diagnostic microbiology. And we asked these two individuals to help us select the expert panelists. And in total, we had about 27 experts on this panel. And we also had our president, Dr. Patel, Melissa Miller from UNC on the planning committee, as well as a number of other folks who were literally trying to help us put this together in literally 3 weeks' time. And the questions that we wanted the panelists to address were what science needs to get done urgently. Here our focus when we charged the panelists was we want to know what we need to know, not the presentation of past research results, but rather for them to focus on the areas of diagnostics and therapeutics, as well as the whole issue of vaccine developments. And we know that there has been a tremendous output of research results from our colleagues in China and from across the globe. But we thought that what this global summit might be able to help us do is have a thoughtful distillation of the perspectives of these experts such that we could come up with a series of coordinated global research priorities. We also provided the panelists the WHO roadmap on COVID-19, which curiously was developed in 2016, but rather than naming it COVID-19, it was Virus X. And as you can imagine, as we began to work through the planning documents and began to think about things, we had lots of discussions. And one of the interesting things was we really wanted to understand how we could help marshal the efforts of the entire scientific community and how to coordinate those activities. And what better place to do that than through the American Society for Microbiology. And so we began to assemble that. And we also could not have a discussion about research without addressing the conversation about how might the research be funded. We all know this is going to take money, but rather than considering where the funds would come from, which agency, which nation state, we really wanted the discussions to focus on how funds might be mobilized quickly, because we appreciate that our patrons, the taxpayers of the respective nation states who are funding the science, really want solutions right quick. They don't want us to throw the grant over the transom, wait 6 months before it's read, and then another 6 weeks before it gets scored, and then another 6 weeks before the funds are assigned, and then another 6 weeks before they are made available. And so we really wanted to get things going. So what did the summit teach us? The short answer is in the 6 hours that we had this conversation, and I should point out it was all done virtually, and surprisingly, the technology worked. We had investigators from Australia who were delightful. They got up. They were up in the middle of the night, and it was 4:30 in the morning when they were talking about vaccine development and some of the other interesting things. And so we just learned a tremendous amount. So I'm going to give you some of the highlights that I picked up just listening to these four folks. We all know the big one, that this virus is highly contagious, and that's owing to the fact that the human race is naive. We have never seen this virus before. And one of the things that I had yet not stumbled upon, but Vincent or Elio probably has seen, is this great website, nextstrain.org, where you'll learn about the over 1,467 genomes that have been sequenced to date. And what this website provides you is it shows you the molecular phylogeny of how this virus is changing. And one of the most remarkable data elements I gleaned is that it's estimated that this virus is only undergoing 26.7 substitutions per year. And Vincent, isn't this virus rather large for an RNA virus? And it's really remarkable that with only 26.7 substitutions, this virus isn't going to change its spots all that much. So we're hopeful that once we develop a vaccine, it will actually be relatively good for a long period of time if they pick the right determinants that aren't subject to hypermutation or things along those lines. So the therapeutics discussion was probably the most interesting to me, as well as the diagnostic discussion. So I'm going to start with diagnostics. Some of you may have read in the popular press that Mount Sinai is in the process of deploying a blood test that measures immunity in recovered COVID patients. This discussion was led by the virologist from Mount Sinai who is leading the team, Florian Kramer, at the Icahn School of Medicine at Mount Sinai, who has developed a serological enzyme-linked immunosorbent assay, or ELISA test. And this test simply asks the question, do you have the presence or absence of antibodies against the coronavirus in your blood? The other thing that was appreciated is that this virus has never been in patients ever. They have gone back and looked at serum banks and have found that the human race has been effectively naive to COVID-19. We have not seen this virus. No one has cross-reacting antibodies against it. So the serological test that is being developed is hopefully going to be very specific. One of the advantages of the serological test is that what Dr. Kramer pointed out is you can pick up in as little as 3 days post-symptom onset as to whether or not an individual has been exposed to COVID-19. And one of the highlights, and this then dovetailed into the therapeutics conversation, is that as people recover from COVID-19, that their serum, which is now considered to be hyperimmune, could then potentially be donated, purified, and then given as convalescent plasmid to help the very sick COVID patients that we're seeing in our intensive care units, to hopefully help them recover. And I'm hoping that it will enable them to get out of hospital much more quickly. The other thing that the serological test affords us is it allows us to determine the true denominator of how many people have been exposed across our nation and across the globe, providing an accurate picture of how many people have been infected and give us a better handle on the case fatality rate, as well as, providing us an opportunity to look at our healthcare workforce, who right now, the only way they can return to work is if they have 2 negative nucleic acid tests that demonstrate they no longer have viral nucleic acid in the specimen that they are using to assess whether or not that individual is still infectious. In addition, this serological test will also allow us to identify convalescent donors so that we may ask them if they would please donate some of their plasma to potentially save other potential plasma donors that are out there, effectively force multiplying. And that brought us to the therapeutic category, where we heard some exciting news from Shmuel Shoham from Johns Hopkins. And Dr. Shoham has been involved with the Academy Chair, Dr. Arturo Casadoval, who is also at Johns Hopkins, where they had submitted an investigational drug application to use precisely convalescent serum from COVID-19 patients to treat the very ill. The rule of thumb that Dr. Shoham socialized with the panelists was right now, before they get into full-scale clinical trial, is it was hypothesized that 1 bag of convalescent sera would be useful to prevent disease, and 2 bags of convalescent sera would be required to treat the disease. So with each passing infection and recovered individual, we, of course, have an increasing pool of convalescent serum. We also heard some of the in vitro work from Ralph Barrick's group on the success of remdesivir. Ralph was unable to join us. He has been, of course, on TWiM a number of times during this pandemic. And remdesivir, while it has good utility in treating COVID-19 or SARS in some of Ralph's animal models, this is an IV-based medication. But Vincent Menachery from the University of Texas medical branch said that for remdesivir to be its most effective, it was required to be delivered to the exposed individuals early in the course of the infection. So remdesivir, we have to know when you have been exposed in order for it to have its full beneficial effect. As some of you may know, there are clinical trials actively going on throughout the United States evaluating the effectiveness of remdesivir in the most severely ill patients. And so there was some speculation amongst the panelists as to whether or not remdesivir was going to be shown to be effective or not. Becky Dutch, another virologist from the University of Kentucky, shared with us the news of a new drug, E91, that is in the process of in safety trials. It's another nucleoside inhibitor similar to remdesivir that's in safety trials, but in contrast to remdesivir, which is an IV-based medication, this one is available as an oral formulation. But this is but one of 100 potential therapeutics that are being evaluated against this coronavirus. We couldn't have had the discussion without asking about hydroxychloroquine and azithromycin, and we asked the fundamental question of how those 2 drugs, the hydroxychloroquine, recall, is an anti-malarial medication, and azithromycin is an antibacterial medication that specifically inhibits protein synthesis in bacteria. We were wondering how mechanistically this could control viral replication in patients. The expert panelists had a number of speculative points, but no one had the gold standard answer. In the prevention category, we then transitioned to vaccine, We heard that vaccine development is underway. We heard from our colleague in Australia, Kanta Subaro, who's at the Peter Doherty Institute for Infection and Immunity. And she, during her time at the National Institutes of Health here in the United States, evaluated SARS, and she was pretty well familiar with the vaccines in the pipeline that are underway and is hoping that the vaccines that are currently being trialed will actually give us some good information. But then, like all vaccine discussions, it morphed into an interesting take. How are we going to make the vaccine available? How is it going to be produced in a sufficiently large quantity? How many countries have the ability to produce vaccines in tissue culture versus whether or not it's going to be another type of vaccine? So the consensus is we should have an ethical discussion about how we can best make this vaccine available across the globe. And so look to another COM summit that will specifically address the whole ethical decisions of how therapeutics are distributed as well as how vaccines are distributed. And then that finally took us back to diagnostics, where we talked at length about the nucleic acid test. We were very excited, the panelists were, that the private sector is beginning to step up. As many of you know, New York State's health department has stood up another number of nucleic acid tests, as have the hospitals in the greater metropolitan New York City. And there are a number of private sector companies that are standing up nucleic acid tests in order to get them into the hands of the public health authorities so we can get a handle, going back to our initial discussions here earlier, about when can we all return back to work. So the takeaway from the diagnostics discussion was diagnostic tests need to be leveraged to help during this pandemic, and we need to ensure that they are standardized. Quality testing is available across our country. And again, we're looking at under-resourced counties in the United States. New York City is, of course, very fortunate in that they have another number of fine institutions that are capable of doing these high complexity testing. Dr. Patel mentioned that it would be ideal if we had a low complexity CLIA wave test that could be distributed en masse, but we unfortunately don't have one of those tests out in the marketplace as yet. The diagnostics group actually got their act together before the discussions even took place, and they have submitted a manuscript to MBio that is going into rapid publication. And so you'll be able to see a discussion in MBio as to the value of diagnostic testing, when, whom, what, and how often to test, that was authored by the panelists who were members of the diagnostic discussion group. The bottlenecks we also talked about. There's an increasing number of institutions shut down across our nation, and they're turning off all their non-COVID-19 related research. And so people are naturally trying to pivot to COVID-19. So we talked a little bit about how those individuals could be harnessed. And another discussion was, as this pathogen, COVID-19 or SARS-CoV-2, is a biosafety level 3 microbe, the biosafety level 3 capacity in the United States is a finite resource. So how can we help crowdsource or harness excess BSL-3 or higher capacity across the nation? Much in the same way astronomers apply for time to use the Hubble Space Telescope or any telescope that's terrestrial based. And then Ben Hurley from Mount Sinai also volunteered that he has been in discussions with Fred Hamprick, and he wants to organize a community of leading machine learners / data scientists and bioinformaticians who have all volunteered their services to help with data analysis ranging from the mundane, please count the particles in this image, to actually doing very sophisticated models. And all of these guys who have volunteer, they are doing it all pro bono. So then this morphed into the discussion of how ASM could help facilitate and coordinate these activities. We also, weaving through all of these discussions was the funding thread. And we heard from a number of individuals, Joanna Sischler from the University of Illinois, who has been serving as a program officer at NSF during this past year. She was talking about how to leverage the NSF investigators. And recall, NSF is statutorily barred from doing health sciences research, but they do have the ability to model and do other things. And so they were looking to see how they might assist the National Institute of General Medicine Sciences and, of course, the NIAID as well. So it was a really rich and fascinating discussion that we had on Monday. It was followed up with a press conference on Tuesday morning where there were a number of reporters from the major news outlets, including CNN, the New York Times, the Associated Press. And so I think COM should be proud that we assembled this august body of scientists, and the discussions were rich. And one of the things that the society is going to make available is you can go back and listen to the entire conference. We're going to post it online as soon as they do a little cleanup of the audio. So that's it, Vincent.
Okay, very good. So listen, can I ask you a question from one of the items that Michael talked about? I don't know how to insert it, but when you talk about antibodies and about convalescent serum, why not monoclonals? Can you identify the antibody and make monoclonals out of it?
Monoclonals were indeed discussed. We brought that up. There are a number of monoclonal candidates, and they are moving those to platforms as well.
Okay, very good. Okay, let's see.
That takes time. You can give convalescent sera right now for serious patients, according to the FDA.
Yes, and that's what Arturo is going. Our Dr. Casadoval has been a whirling dervish trying to line up blood banks. He's contacting his colleagues across Maryland and New York. For many years, Dr. Casadoval was in New York City. He has deep contacts in the New York City hierarchy to get access to the blood bank. So they're very excited about being able to get this into patients as quickly as possible. So he really went to the mat to get this IND done. We talked about some of the chaos associated with submitting things to the FDA. It requires a paper copy to be mailed to the FDA, and except nobody is at work anymore, so it caused a bit of a kerfuffle. You know, people had to go in and put it into the FedEx box, and then it had been sent to the FDA, but they managed to get it done.
Great. It's very impressive. So you guys want to do a paper, or?
Yes, sir.
Because I could do this in 15 minutes.
All right. It looked like, I read it, and it was a cool paper.
It's a very cool paper. This is published in Cell Host and Microbe. Paper's Salmonella secreted effector, SARA, STEE, mimics cytokine receptor signaling to activate STAT3. Now, don't get scared. That's a complicated title, but I'm going to make it simple, or simpler. This comes out of Duke University.
these people want to get their act together about what to call the different compounds, the different proteins. It's a mess.
Oh, yes.
The nomenclature is a mess.
I agree. There's a lot of different names for the same thing, right?
Absolutely. Well, I made a cheat sheet. I had a glossary, and I made a cheat sheet. But I had to do that.
Well, I'm going to make it simple, so you won't need a cheat sheet.
Oh, thank you.
So, Salmonella, important pathogen, 150 million diarrheal illnesses a year. You know, that kind of puts SARS-CoV-2 in perspective. Yeah,
we only have 500,000, and it has shut the planet down. Yet 150 million people with diarrhea, nothing.
600,000 cases of bacteremia, 25 million cases of enteric fever every year, the highest morbidity of any foodborne pathogen. Part of the reason why it's so successful is that it injects into cells effector proteins that modify the cell so that the cell now is a wonderful environment for itself. I love how they say this, the first sentence, bacteria masterfully co-opt and subvert host signal transduction. So that's what this story is about today. So this Salmonella enterica has 2 type 3 secretion systems which deliver proteins. These are called effector proteins. They're basically proteins that have an effect on the host cell. They manipulate the physiology. They allow the invasion of the bacteria. They establish a niche so the bacteria can multiply. And this story is all about an amazing way that Salmonella enterica modifies the host defenses. So they're basically, I'm going to tell you a story of how a Salmonella protein called SARA, S-A-R-A, and this is the one Elio was complaining about because it has 4 other names in the literature, but we'll call it SARA, promotes an anti-inflammatory response. So what does that mean? Well, when you're infected with a bacterium or a virus, there are proteins in cells, in our cells, that sense the infection, and they mount an antimicrobe defense, which involves production of cytokines and chemokines, and these recruit immune cells into the area to get rid of them, but in the process, of course, it causes inflammation. You get swelling and redness and pain, but in the end, the idea is to cure the infection. And one major way that this happens is by ligands binding to receptors on cell surfaces. And among the cytokines that are produced is one called IL-6, interleukin-6. This is what we call a pro-inflammatory cytokine. It's made early on in virus and bacteria infection to help recruit all these mediators into the area and help clear the infection. IL-6 in itself can bind a receptor on host cells, and that binding induces the production of more cytokines, but then it also induces the production of regulators that turn off the pro-inflammatory cytokine response. That's because you don't want to have inflammation forever, because it's not really good for you over the long term. You know, that's what a flu-like symptom. It'll kill you.
It can kill you.
That's right. Too much inflammation. Flu-like symptoms are inflammation. And in fact, a lot of the pathology of SARS, COVID-19, is due to inflammatory overexuberance in your lung. And in fact, some of the treatments that are being considered are using a monoclonal antibody to IL-6. Treat patients with that to try and dampen down the inflammatory response. Because IL-6 is a bad well, it's not bad, but you can only take so much IL-6. Too much is not good. So built into the IL-6 pathway is this overriding pathway where it's called SOCS, S-O-C-S, suppressors of cytokine signaling. It's a way to shut off the pro-inflammatory cytokine signaling. And so what Salmonella, SARA, does is to completely turn on the suppression part of that pathway so that all the inflammatory responses are suppressed, and that's good for the bacteria then because there are no immune cells coming in to get rid of it, and it can multiply. That's very devious. So it takes advantage of an existing pathway for suppressing inflammation and turns it on constitutively to its benefit. So here's how it works. There's this protein on the cell surface. It's a receptor for IL-6. The ligand, which is IL-6, binds to this receptor, and that binding causes phosphorylation of the cytoplasmic portion of the receptor, which then recruits a cell protein called STAT3. STAT3 is phosphorylated. It goes in the nucleus and turns on the transcription of genes that are involved in anti-inflammatory responses. So this SAR-A protein, the authors looked at it and they said, this has a sequence. It has a 39 amino acid sequence that looks an awful like the cytoplasmic domain of this IL-6 receptor. And so they find out eventually that this part of SAR-A actually will bind STAT3. So remember, STAT3 is normally binding the cytoplasmic domain of this IL-6 receptor. Now this bacterial protein SAR-A is binding it instead. That is going to now rewire, if you will, the response to be overproducing the suppressors of inflammation. So by just looking at the sequence, they identify this 39 amino acid domain. They do some experiments to show that the protein will bind, the SAR-A protein will bind STAT3, and that suggests this mechanism where the SAR-A can functionally substitute and actually does a better job at turning on this pathway than the normal pathway does. It turns out that this SAR-A is phosphorylated at a specific tyrosine residue, and they show by knocking out that amino acid that phosphorylation is important for the activity of SAR-A, that is binding to STAT3. SAR-A binding to STAT3 would then lead to phosphorylation of STAT3 and would go in the nucleus and turn on all the anti-inflammatory pathways. So that phosphorylation is crucial for that activity. They show that in a number of very interesting experiments. This SAR-A protein binds with very high affinity to STAT3, even more high affinity than the cytoplasmic domain of this cell surface receptor, the IL-6 receptor that we've been talking about. So it's evolved to be really very good at this diversion of the pathway. So they want to next know how this works. And so they take the SAR-A protein and they do what's called a discovery screen, where they make the protein and they add it to cells and they ask what other proteins are binding to it. And they identify them by mass spectrometry. And among the proteins they find are a couple of other protein kinases in the cell. They're called GSK3 alpha and GSK3 beta. Names are not so important. What's really interesting is that they eventually show that the activity of one of these kinases is absolutely needed for the activity of SAR-A. That is, the ability of SAR-A to bind the cytoplasmic port, to bind STAT3 and to phosphorylate it. And apparently GSK is the kinase that is phosphorylating STAT3, because SAR-A doesn't have any kinase activity on its own. So here we have this model in which the bacteria injects into the cell SAR-A protein. It binds to this cellular kinase GSK3. It then phosphorylates SAR-A, and then that results in binding of STAT3. STAT3 gets phosphorylated, probably by GSK, and it goes in the nucleus and turns on all the anti-inflammatory genes. So normally, again, to summarize this, the IL-6 would bind to the cell surface receptor. The cytoplasmic domain would be phosphorylated. That would bind STAT3. STAT3 would be phosphorylated, would go in the nucleus. But now this bacterial protein, SAR-A, is taking the place of the cytoplasmic domain. Itself binds this kinase GSK3, and it binds with even higher affinity to STAT3 than does the cytoplasmic domain of the IL-6 receptor. And so, I must
say, they do not explain it very well in the graphic abstract, because it doesn't really show that. So I get another beef with this paper.
Well, but that's my take on it, right? That this is what's happening. SAR-A is diverting STAT3, turning it on constitutively so now the cell is only making anti-inflammatory proteins. There are cytokines that are anti-inflammatory that turn off the inflammatory response, and this is good for the bacteria, right? Because inflammation would clear the infection. Sure. I think this is just beautiful. The fact that it exists and they're able to sort it out is quite interesting. It makes you always wonder at evolution how it is selected for this, selected for proteins that have evolved to steal this pathway. So you no longer need a cytokine to activate this pathway. The bacterial protein does, and it makes an environment that's favorable for the multiplication of the bacteria. Anyway, did you like that story, Michael?
Oh, I disagree with Elio. I think the graphical abstract really does it justice. And at the end of the day, the take-home message is Salmonella gets inside the cell and turns off the host ability to effectively dispatch that infected cell with the Salmonella. And the net consequence is you have increased replication of the Salmonella inside the cell, and that's why it's such a phenomenal facultative intracellular parasite. And I think if we looked for the same behavior in another pandemic, and that would be pneumonic plague, we would probably see, I would lay even money, that pneumonic plague probably similarly downregulates IL-6 akin to what we see here to effectively being anti-inflammatory, because bacteria rarely invent things multiple times. Mm-hmm. And this is really, I think, appropriate because one of the things that I didn't go into when I was talking about some of the virology that was discussed at the summit is that the SARS-2 virus has immune modulating genes, and that's effectively what we see here with Salmonella. This is an immune modulation that is enabling this bacteria to cause 150 million diarrheal diseases.
Yeah, I agree. Very cool story.
It is.
I thought everyone would like a little distraction from viruses, so there you go. Let's read.
That was nice.
Let's read a couple of emails. We have some here that are nice. One is from Amir, dear TWiV team. My name is Amir. I'm an Israeli PhD student studying in Jena, Germany. We haven't got a real winter this year, so it's not too cold, around 5 to 10 degrees Celsius. Thanks for all the hard work. I've been listening to you guys for a few years now, and you really bring out cool science that I wouldn't otherwise hear about, so thank you. A question came up at our department seminar that sparked a discussion I wanted to share. A novel method was presented for medical diagnostics using Raman spectroscopy, and somebody asked whether all infections are single species. The speaker was stumped. Fair enough, he's a chemist. So I roll the question to you. Are infections generally single species? I'm sure the answer, as always, is that anything is possible. But what is the norm? I remember most infections are monocultures except for rare cases, but please enlighten me. I want to add that I work in the Department of Chemistry, so most people really have no background in microbiology at all, and I studied agriculture, so my medical training ain't too sharp. Thanks again, Amir.
So I'm going to answer on behalf of all the dentists in the world. The answer is no. There are a lot of mixed infections, especially in the oral cavity. The best example is good old trench mouth, which is acute necrotizing ulcerative gingivitis, which is caused by streptococci, fusobacterium, and oral treponemes. So those three organisms get together and they make literally a real stink in your oral cavity. The person who has acute necrotizing ulcerative gingivitis, or ANUG, the minute you ever see that patient, you will remember that odor for the rest of your life.
I think other infections are... Go ahead, Elio.
I should also add, I had a brief stint of teaching vet students, and it turns out that a large number of vet infections are multi-species, large numbers. So maybe in humans it's a little bit less, but like Michael says, it ain't nothing. They are very important.
Same with viruses.
They're certainly.
I mean, a lot of these individuals who have COVID-19 actually also have influenza, for example. Very common.
Well, yeah, what is it, 5% of the cases?
Yeah, it's very common to have multiple viral infections. It's probably not the rule, but it's more common than we think because we never used to look, but more when you look, you find. Yeah.
And that's certainly the case in prosthetic joint infections.
Yeah.
If you're in a clinical lab doing microbiology, the overwhelming number of things you find are single-species infections. They are more common generally in humans.
Okay.
It's not, but the multi-species infections are not rare, and they're terribly important.
Yeah. By the way, Amir says a note on Jena. Jena is the home of Zeiss, one of the best companies for microscopes in the world. Carl Zeiss has pretty much invented the modern microscope together with Ernest Abbe, who discovered the resolution limit, and Otto Schott. They wanted to make science more accurate, reproducible, and standardized worldwide. This is a huge...
They discovered the condenser. Their big contribution was to make a condenser for a microscope. The thing is underneath.
I see.
Underneath the object.
And here I thought Otto Schott invented the shot glass.
This is a huge part of the city's culture, with streets and buildings named after the three and the local sports teams, as well as statues presenting Zeiss with his microscope, Abbe's equations, and a full optics museum. I went to Jena once years ago. I remember there's Goethe University there, I think, if I'm not mistaken. All right,
we should get somebody from Zeiss to come on and talk about advances in Numarsky optics and some of the other cool things in visual microscopy for one of the upcoming TWiMs.
Yeah, we could do that. I'm sure they'd love to come on, right?
Yeah.
We have an email from Bob who writes, the world of bacterial outbreaks is happening at the same time as COVID-19. Apparently, there's an outbreak of listeria caused by enoki mushrooms.
Oh, my God. Really?
One of Elio's favorite things on the planet, mushrooms.
And listeria, as you know, grows under refrigerated temperatures. It's quite happy at 4 degrees, and it is a nasty little foodborne pathogen.
Yes.
Enoki mushrooms, by the way, are cultivated. So there is a source for this to happen in the place where they're grown. They're not harvested from nature. In fact, the odd thing, and this is an aside, if you find the same fungus in nature, which it's called a winter mushroom, because it grows on trees like elms in the wintertime, it's completely different than enoki. It's a brown little mushroom with a cap and a stem, and it looks nothing like enoki.
And so
that's kind of fun.
The fact that it's farmed means probably some contaminated water was used, right?
That's right.
Contaminated water.
Mm-hmm.
Yeah. If you want to know more about enoki in our blog, we're going to have a piece about it very soon.
Okay. All right, and our last one is from Matt. Kind greetings. I am new to TWiM. I just listened to my first episode, a fascinating show covering topics ranging from a discussion of tripartite horizontal gene control in mealybugs to hospital floors being a hotspot for coronaviruses. Thank you. My question is about hygiene. The general public is correctly told that diligent hand washing is helpful in reducing infection risk during any outbreak. Anecdotally, for the last 8 years, when I have been inadvertently exposed to a very sick person spewing virions with great force near me, I have proceeded to diligently wash my hands afterwards. Once my hands are clean, I wash my face, nostrils, tear ducts, lips, ears, and general facial area for 30 seconds. I seem to get less sick than my peers, and when I do become sick, it's less severe. I surmise this could be related to consistently lower viral loads delivered to the principal portals of pathogenic entry into my body, namely my facial mucosa. I have a few questions about this procedure. Do you think a study using harmless cauliflower mosaic virus is warranted to see if this practice could be meritorious? If so, who might fund it? 2, how could this procedure be made safer? I note facial touching and face washing is still routinely practiced among the general population during this COVID-19 pandemic when showering daily. Thank you, Matt from San Francisco. So it sounds like to me, Matt, that you're very fastidious about washing, and that's probably helping you to be more free of infections than your peers. I think there's a relationship between the two for sure.
A lot of folks have looked at using something like cauliflower mosaic to see how it moves through hospitals. I think we talked about that on a TWiM when we were discussing phage, and they put the MS2 virus, if I'm remembering right,
that's right,
on the doorknobs, and they watched it, and it ended up in the coffee room most often. So those studies have been done in the past, not using cauliflower mosaic, but I think it was MS2 virus. I'll have to go back and look at the old show notes, but those studies have been done, and in fact,
but not on touching your nostrils and your face and your eyes, etc., which is what is being proposed here.
Well, what he's proposing to do, but in the master question list published by the Department of Homeland Security Science and Technology Division, they have a master question list for COVID-19, and that's one of the questions on the list is what is the infectious dose for COVID-19? So, getting the cauliflower mosaic virus, I'm sure someone is going to look at how important hand hygiene is in controlling these viruses.
Yeah, I think, Matt, if you have a look at that literature, you'll find who supported it. Michael, who would support studies like that, do you think? NIH? It's not NSF.
NIH. It's Fundamental Basic Science Research, and that's NIAID.
But maybe you could get a hand-washing company to be interested,
Yeah.
Not hand-washing, but soap.
Somebody like Perel or Gojo or any of the alcohol-based hand hygiene companies. If it's, I'm forgetting, is cauliflower mosaic an envelope virus? I don't remember. I'm only a virologist on television.
On television? No, it's not.
They gave the press conference. They gave me many titles. I was an epidemiologist. I was a virologist, and I was also a microbiologist. So the press gave me many different. They conferred many different titles on me during that.
It's a non-envelope virus, Michael. It's pretty hard.
Okay, thank you. So it's harder to kill.
It's harder to inactivate. Yeah. All right, thanks for your letters. If you want to send some to us, TWiM@microbe.tv. You can find all the show notes at asm.org/TWiM. If you like what we do, consider supporting us financially, microbe.tv/contribute. Today's hosts here on TWiM from a really nice blog. You should go check it out, Small Things Considered. You just search for that. You'll find it. Elio Schaechter, thank you very much, Elio.
My pleasure.
And from the Medical University of South Carolina, Michael Schmidt. Thank you, Michael.
Thank you, Vincent.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank ASM for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega, who's going to earn his pay editing this one.
Oh, wow.
We're sorry, Ray.
Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.